Good morning, grade 11s. So to continue with the structure of the urinary system and the production of urine, I take it from the fact that none of you questioned, other than one student, anything about the nephron and the kidney, that you actually all understand everything that I've dealt with so far. As I said to you when I started this whole section, it's going to seem very bitty and PC, and it's going to be that I'm going to describe something very superficially, then I'm going to go backwards and forwards and backwards and forwards, because it's difficult to understand the process unless you understand the structure. But if you don't know a little bit about the process, the structure seems completely meaningless. So that's why it goes backwards and forwards and backwards and forwards. But I promise you, it will all fit together in the long run. Okay, so now I'm going back quickly to talk again about the nephron, just to make certain that you understand it, because you're not going to understand how urine is made if you don't remember the structure of the nephron. So very simplistically, on the drawing on the left side, let's have a look. Remember, each kidney is made of hundreds of thousands of nephrons. That is what makes the composition of the actual kidney, is all these little nephrons lined together. So this is one nephron, highly, highly magnified, showing you the different parts and showing you where they sit relative to everything else in the kidney. So what you can see is there on the drawing is a little renal corpuscle or a Malpighian body, same thing. And then the rest of the nephron is the renal tubule. So the renal tubule, remember, is proximal convoluted tubule, loop of Henle, so descending limb, ascending limb, distal convoluted tubule, and collecting duct. So remember that these stripes within a pyramid are in fact made up. The bigger stripes, you can't see the small ones really, and you can't even really see the bigger ones to that clearly. You don't see it as being striped like this. But the pyramid is made up of the collecting ducts and the loops of Henle. Loops of Henle are much finer than the collecting ducts. Okay, so within the cortex or outer layer of the kidney, Malpighian bodies, proximal convoluted tubule, distal convoluted tubule. Within the medulla, and in fact specifically within the pyramids of the medulla, loops of Henle and collecting ducts. Okay, all right, so let's have a look back at this one again. So putting into perspective on the drawing, remember within the cortex would be a renal corpuscle, proximal convoluted tubule. Then the tube straightens out and forms the loop of Henle. So descending limb of the loop of Henle, ascending limb of the loop of Henle. Don't obsessively know about descending and ascending, but they're so obvious. Okay, distal convoluted tubule. Distal because it's distant in terms of where it is relative to the renal corpuscle, and convoluted because it's windy, and tubule because it's tubular. And then all the little distal convoluted tubules in that region would join together and form a big collecting duct. And remember that the collecting duct then goes to the calyx. So remember that the calyx is a hollow extension of the renal pelvis, and it surrounds a number of these collecting ducts. And so urine would pass down the collecting duct into the hollow of the calyx, into the renal pelvis, which is a hollow basin, into the ureter, into the bladder, and finally to the exterior through the urethra. Okay, all right, so quickly revising a look at the blood system. I told you don't learn this terminology down here, the arcuate, artery and vein. You just need to know that this is a branch of a branch of a branch, and you don't even have to know how many times there are branches there, because I've just made it up, of the renal artery. So this is from the renal artery, and this is going towards the renal vein. So here's a branch of a renal artery, and a tiny, tiny artery. So it's called an arteriole because it's very small, and because it's taking blood in towards something, it's called afferent. So an afferent arteriole leads into the glomerulus, and what it does is it divides into a whole network of capillaries, which is called the glomerulus, which is inside the cup of Bowman's capsule. Those capillaries then rejoin and form the efferent arteriole leading out. Now, you know from the previous lesson that the bore, in other words, the diameter of the lumen of the afferent arteriole, is greater than the bore of the efferent arteriole. And this diagram certainly doesn't look like that. But anyway, what you're going to remember is if you get a diagram and you asked which one is which, firstly look at the bore or the structure. Now, the problem is that if you're looking at the external structure, you can't see the bore. But what then you have to look at is follow it. If this one leads to the peritubular capillary network, as it does here, this is the efferent arteriole. Because remember that the efferent arteriole divides into a network of capillaries that closely surround the rest of the tubule. And those capillaries all rejoin and form a little venule, and that venule joins other venules and eventually will lead to the renal vein leaving the kidney. Okay, right. And then this was, I put this slide in because I wanted to show you that it's not always that the, as this one does, there's the renal corpuscle and it leads into the proximal convoluted tubule, and this left side here is the loop of Henle descending, and then here on this right side is the distal. Remember I said to you, sometimes you'll get a drawing where the two parts of the tubule have twisted over each other. And I said, be careful, follow from what you know to what you don't know. So start at the glomerulus and follow. This is proximal. And the proximal here in this drawing goes behind the distal. And so now the proximal is on this side, and loop of Henle and loop of Henle, and then the distal crosses over again. And there we've got the distal and the collecting duct. So be careful, because if they say to you, identify this part and describe its function, and you've got the wrong part, then you're going to lose all those marks on function. Okay, so I'm just telling you to be careful. All right. Okay, let's quickly go back and look again at the renal corpuscle, or the Malpighian body. Remember, Malpighian, capital M, and it's GH. There's no H here. Okay, so quickly revising the tissue types in the different regions. So remember that capillaries always have a layer of squamous epithelium as their wall. By definition, a wall of a capillary is a single layer of squamous epithelium. So it is cells lying on a basement membrane. Remember that the cells in the glomerulus have got tiny little pores between them, and in fact also going through the cells. Because they're very tiny pores, those pores are called micropores. So it will be blood in the capillary, then it will be a layer of cells, and then it will be a layer of basement membrane. Okay. Then looking at the wall of Bowman's capsule. So we're only interested in this inner wall here. Okay. Remember, I said to you that's also squamous epithelium, but those cells are modified. They're modified, they're not normal squamous epithelium cells. They're called podocytes, and they look a bit like starfish. and they also rest on a basement membrane, but I'll go back to that. So those starfish-shaped cells, in fact, don't lie flat. They curl up around the capillaries. You don't have to obsess about that, but if you look at a picture where those little podocyte cells are curled up around the capillaries, that is what they do. Okay, and then there's a layer of basement membrane. No, the basement membrane is first. Okay, so going. Blood in the capillary of the glomerulus, then a layer of cells, then the basement membrane of that squamous epithelium of the capillary, then the basement membrane of the squamous epithelium of Bowman's capsule, then the podocytes. So it is cells, basement membrane, podocytes, cells. Okay. Now, do you think that just now I said the basement membrane of Bowman's capsule underneath, but it's not. It's above those cells. I hope that makes sense. Okay. You're right with micropores. So let's have a look. The way in which this functions very simplistically goes. The bore of the afferent arteriole is bigger than the bore of the efferent arteriole. Are you looking at the drawing? Okay. Therefore, blood coming into the glomerulus and blood leaving the glomerulus, different volumes. More blood can come in than can go out. Because more blood comes in than can go out, it sets up a pressure in the capillaries of the glomerulus. That means that all the small molecules, so amino acids, glucose, some salts, some water, not all of the water, there's not enough time for all of the water to go through, and certain other tiny, tiny molecules will pass through the micropores, pass through the basement membrane, the slits of the podocytes, and end up in the space of Bowman's capsule. That process is called ultrafiltration because it is of very small molecules. So you'll see it referred to as ultrafiltration or glomerular filtration. Either one is okay. I think ultrafiltration is nicer because it reminds you that it's of very small molecules. Now, I just quickly need to talk about what is the filter here. Relative size, the micropores are much bigger than the molecules. So it's not the micropores that are the filter. The slits between the podocytes are also relatively big compared to the molecules. So what is the filter here is the two layers of basement membrane. The basement membrane, those fibers of the basement membranes are so close together that they act as a filter. Only very small molecules can pass between those fibers in the basement membrane and go from the blood in the glomerulus to the space in Bowman's capsule. Okay, so it's very important that you know that what acts as the filter is the basement membranes. Okay, so that's ultrafiltration, and the liquid that has passed through here is called filtrate. And that liquid then passes down the rest of the tubule. And remember that the rest of the tubule is very long because this filtration is simply on the basis of size. It's got nothing to do with toxic versus useful. And so the problem is that a whole lot of useful substances filtered through and certain toxic substances remained behind in the blood. I forgot to say that urea goes through there, but it is listed for you a little bit later on. And so the purpose of the whole of the rest of the tubule is sort out the fact that there are useful substances in the filtrate and there are still toxic substances left in the blood. So as the filtrate passes through the rest of the tubule, that is going to be sorted out. Things that are useful are going to be absorbed through the walls of the tubule and go into the peritubular capillary network and be restored to the body. In other words, they'll become part of the blood again and the body can use them. And the toxic things that remained behind in the blood supply here are going to be pushed from the blood in the peritubular capillary network. So the capillaries would be here. Those waste substances are going to be pushed from that blood through the walls of the tubule and into this lumen of the tubule here and become part of urine. And that will be sorted out through the whole of the rest of the tubule. Okay, so remember the peritubular capillary network, that's going to be important. Okay, so this particular slide just shows how the podocytes don't form a flat layer, but they actually curl up and surround the capillaries. And then you can see the slits between the podocytes. And then you can also would be able to see the basement membrane. Okay, all right. I did that all last time. I'm quickly going to revise this because this is also important. So we're looking at the walls of the renal tubule, and remember that the walls of the renal tubule are simply a single layer of epithelial tissue resting on a basement membrane. And that it's different in different regions because they have different functions in different regions. So look at the pictures. If you look on the micrographs, these little colors here, that's a key to the color on the region on the tubule. So mustard is just a proximal convoluted tubule. Green is distal convoluted tubule, etc. So let's go. Okay. Proximal convoluted tubule, single layer of cuboidal epithelium with microvilli and many mitochondria, which you can't see because this is not an electron micrograph. Okay, it's a light microscope that was used for this. So cuboidal epithelial cells, many microvilli, many mitochondria. And I'll show you that on an electron micrograph now. And how do you know that the cells are cuboidal? You learned last year, in cuboidal epithelial tissue, the cells, the nuclei are spherical. So look, they're little round balls. Okay, I'm going to go down and compare it with squamous epithelium. So here's some squamous epithelium, and you can see that the cells are not cuboidal. Okay, the nuclei are not cuboidal. Making these errors, sorry guys. All right, so proximal, cuboidal, microvilli, mitochondria. Loop of Henle. Loop of Henle is squamous epithelium. So here's a loop of Henle, and here's a loop of Henle, and here's a loop of Henle, and the cells are very flattened, squamous epithelial cells. Okay, then increasingly going up this ascending limb, there is a change from squamous to increasingly more cuboidal. So it goes from very flat to less flat to less flat to cuboidal. And so that's distal convoluted tubule that you can see here. And nice clean inner surface, so no microvilli. And this would be quite low down here because it's not entirely cuboidal, it's still a little bit flattened. And the cells are cuboidal. Okay. And then the collecting duct is very cuboidal cells, in other words, nice spherical nuclei, clean cut inner surface, so there are no microvilli. And look how much bigger the lumen here is than the top one, the proximal or even the distal convoluted tubule. And that's because the collecting duct is made of a number of distal convoluted tubules joined together. Okay, so the lumen has just gotten bigger and bigger. Okay, So let's have a look at an electron micrograph. And this would be a transmission electron microscope because it's not 3D, it's a thin slice. And here you can see, and I'm going down to the bottom right-hand corner because this is an awesome proximal convoluted tubule. So you can see the microvilli. Look at them. Okay, so it's not a clear-cut inner edge. I'm going up to the distal above it, clean-cut inner edge. So no microvilli, going down, microvilli. Now I'm going up to the top left-hand corner. Look at the many, many mitochondria you can see in these cells here. And there's a little spherical nucleus, which tells you it's cuboidal, and there's a spherical nucleus as well. Okay, this one is absolutely perfect. Look at the spherical nucleus, tells you it's cuboidal. Look at the microvilli, tells you it's proximal convoluted tubule. And look at the many mitochondria, proximal convoluted tubule. Okay, all right. So the 3 stages of making urine are ultrafiltration, also known as glomerular filtration, but I'm happy for you to just learn this term. Tubular reabsorption and tubular secretion. Now, I don't know if this is showing for you. Something's just popped down here as I move the cursor up. Absorption, be careful how you spell it. And I told you this when we were dealing with nutrition. There's a P here. Okay, so 3 steps in making urine. Blood that runs through the capillary network in the glomerulus is filtered by the melting in body. Most of the solutes in the filtrate are reabsorbed in the proximal convoluted tubule. Remember, that's because they're useful still. And that process is called tubular reabsorption. As the filtrate passes down through the loop of Henle, there's a continuous exchange of solutes and water within the renal medulla, with the renal medulla and the peritubular capillary network. And I'll deal with that. And then, remember I said to you that certain substances are big molecules and remained in the blood. And so those need to be pushed into the urine. And so they pass from the peritubular capillary network, mainly in the distal convoluted tubule or the collecting duct, into the filtrate. And that is tubular secretion. Okay. So urine then is a collection of substances that have not been reabsorbed during glomerular filtration or tubular reabsorption. Okay. So this shows you nice and clearly on a drawing. So starting at the glomerulus, that is ultrafiltration. Small solute molecules pass from the blood in the glomerulus into the renal tubule. Okay, remember, ultra because it is small molecules, and filtration because it's a separation mechanism. So because there is a pressure in the glomerulus, and remember, that's because the bore of the afferent arteriole is bigger than the bore of the efferent arteriole. More blood can come into the glomerulus than can leave. Because of that pressure, all the small solute molecules pass through the 2 layers of basement membrane and become filtrate. All the molecules that are not small enough will remain behind in the blood. Remember that in the proximal convoluted tubule, a lot of reabsorption occurs. So this is the main region, I'm looking at the text now, of tubular reabsorption. So the ions that are useful to the body, some of the water, all of the nutrients in a normal healthy person who has a normal healthy diet. So all of the glucose and the amino acids will move from the filtrate into the peritubular capillary network. That's tubular reabsorption. Certain substances will go from the peritubular capillary network, some of the toxins, and certain, there's a little bit about the pH balance here, but we'll talk about that later on. Mostly that occurs in this region, okay? And I will talk about that later. So a lot of tubular reabsorption occurs in the proximal convoluted tubule. We'll talk extremely briefly about the role of the loop of Henle in this whole thing. And it's mainly that the loop of Henle, salt molecules pass out of the loop of Henle. And they go into the fluid of the medulla between the nephrons. And that's going to be important later on. But we're going to talk about it extremely briefly. I'm not going to do it in a lot of detail. And then the filtrate passes up to the distal convoluted tubule and the collecting duct. And the distal convoluted tubule and the collecting duct, the main important thing here is the body's able to control how much water it reabsorbs. There's a part in the brain that constantly monitors how much water there is in the blood. If the blood contains a lot of water, in other words, it's quite dilute, and the body doesn't need to conserve water, then the walls of these tubes become impermeable to water, and the water just keeps on going, and out it passes in the urine. If, however, that part of the brain senses that the blood is very concentrated, there's not very much water in it at all, then the body needs to conserve water, can't afford to lose that water in the urine. And so what happens is these walls become permeable to water, and water passes from the filtrate into the blood and is conserved in the body. I'm going to talk about all of these stages in a lot more detail later on. So I know you're going, oh, she went so fast there, I didn't take enough notes. I'm just explaining very briefly to you at this point. Okay, so ultrafiltration, tubular reabsorption. Mostly concerned with pumping out salts, ions, because it's making this fluid here have a high concentration of salts. And that's to do with conserving water. But there's no regulation of water conservation here. We'll talk about that later on. This is mostly to do with conserving water or passing the water out of the body. We're going to talk about that. But a little bit about, if I look up in the text in the top right-hand corner, certain ions being passed through from the filtrate to the blood or vice versa to maintain blood pH. I never knew that the kidneys functioned to control the pH of the blood, but they do. They're amazing. They're incredible. Okay. All right. I'm going to leave it at that. Right.